Hi all, welcome back. We're continuing the talk on plasma membranes. So just continuing cells, the lecture on cells, and diving further into plasma membranes, and then we'll discuss prokaryotics, the simplest cells that we have on the planet, or have discovered on the planet at least. Okay, so plasma membranes, remember after we discussed plasma membranes before, that we talked about this membrane, this kind of sac-like structure that was on the outside of the cell. Okay, that plasma membrane is what we call a fluid mosaic, or it's part of a fluid mosaic model. Okay, what's meant by a fluid mosaic is that it's not rigid, it's flexible. Okay? And the flexibility comes with these lipids, okay, these phospholipids that are making up the majority of the membrane, and the proteins that are embedded in between those layers. Okay, and it is a phospholipid bilayer. So there's actually two layers, and I'll show you a picture here in a second of those lipids and proteins that are associated with it. So when you think of a sphere and kind of think of an object that might be similar to this, I want you to think that it's not a basketball. Okay, so the outside of a basketball is rubber or leather or some kind of rigid structure. It's a tiny bit flexible, a basketball is, but you can't bend it into a completely different shape. Okay? That's not the case. Okay? Cells are completely flexible. It's more like a half-filled water balloon, okay, with little teeny holes in it. But it's really flexible, and you can bend it all over and reshape it quite a bit. And the flexibility will determine its functionality, and we'll talk about that here in a second. But again, those lipids are those fats. So lipids just another name for fat. Those lipids are what we call phospholipids. Okay, so it's got a phosphate head and a fatty acid tail, or fat um, along the tail. Okay, so looking at this is an example. This is a sketch of that fluid mosaic model. Okay, so you have these fatty acid tails and these phospholipid heads. So the phospho or I'm sorry, you have the phosphate heads and the fatty acid tails. Together they make a phospholipid. Okay? We call it a bilayer because you have one on top and one on the bottom. Now, the heads can interact with water, with fluid, okay, and they do so. So out here would be fluid and inside would be fluid. So you can see the heads are pointed towards the liquid and the tails are pointing away because the tails are hydrophobic and the heads are hydrophilic. Okay, and we'll come back and talk about these. Here are those proteins. Okay, so you have one protein here, it's got a binding site on it. If it's got a binding site, it probably means that things can enter the cell or exit the cell. Okay, so these are transmembrane transport proteins. Okay, so things can go in and out. Not all the proteins are associated with entering the cell or exiting the cell. Some are with self-recognition or just recognition by other things. So here you have a protein with a carbohydrate. Carbohydrate's a fancy word for sugar, and that sugar is attached to that protein. This is a recognition protein, so it recognizes, maybe the immune system recognizes this as self-made, so it doesn't attack it. Okay, or maybe other cells recognize it as, this thing can bind with us, and that, so those units are used to recognize. Okay. You can see a bunch of protein scaffolding, long strands in here, the filaments and other things going on, and that just provides some rigidity with inside the cell, and we'll talk about it. It allows for some of the structures um, to not move around as much inside the cell. And then you can see here in this picture one of the key components, and we'll come back and talk a little more about this. But you can see cholesterol is bound in between these phospholipids. And often we talk about cholesterol as being kind of a bad thing. But cholesterol is like a double-edged sword. Some cholesterol is actually very good and it's actually needed. And that's because without cholesterol embedded in these membranes, your membranes would be too fluid. Okay? And they'd actually start to kind of be leaky and have material kind of leak out because the flexibility would be too flexible. So cholesterol provides a little rigidity to it. But you can imagine if it provides a little rigidity, too much cholesterol embedded in these membranes means your water balloon just became a basketball. And now there's no flexibility in that outer membrane. And for things like red blood cells, which are moving through veins and arteries, which might be fairly large, into capillary beds, which are really thin and small, those red blood cells need to be able to squeeze down and squeeze through areas. Okay? And if you have a bunch of cholesterol on it, they can't squeeze anymore. And that's problematic. And we'll come back and talk about those issues later. Okay, so diving back in, polar heads, non-polar tails. The polar heads are water soluble, okay? So they're hydrophilic, and the non-polar tails, okay, are water insoluble or hydrophobic. So here you can see you have this phosphate head, okay, and then it's normally attached to the fatty acid tails by glycerol. So sometimes people will say, well, it's a phosphate head, a glycerol neck, and tails made out of hydrocarbons. Okay, so carbon being these black circles, and the blue circles are hydrogen. So that's what these fatty acid tails are, really just carbon and hydrogen. If they're linear like this and there's really no kinks, they are what we call uh, saturated, saturated fat, okay, meaning it has all the possible hydrogens it could have. If it's considered an unsaturated fat, it'll have like a kink, okay, where the two of the carbons are double bonded to each other. Okay? And those are really kind of the molecule differences between saturated fats and unsaturated fats. Okay, and there's debate on which one's better for you, blah, blah, blah. We're not going to dive into that right now, but they have different properties. Okay, saturated fats, solid at room temperature, unsaturated fats, liquid at room temperature, and that's really comes around from these chemical forming chemical bonds. Okay, so whether or not they have a double bond or straight single bonds. Okay, we'll come back and talk about that later. All right, let's dive in and talk about prokaryotic cells. Okay, so prokaryotic cells, there are two types of cells. Okay? You have eukaryotic cells and prokaryotic cells. Pro means before. Karyotic means nut or seed. Okay? So prokaryotic cells are referred to as before nut, before seed, or before center, okay, or before nucleus. Okay, so prokaryotic cells, they do not have a nucleus. Right? They actually have no organelles. So there's no internal membranes. Okay? Now, I want you to remember this though. Just because they don't have a nucleus and they don't have a Golgi apparatus and they don't have a mitochondria and all these other organelles that we'll talk about next lecture, okay, it doesn't mean they don't have the capability of doing the functions that those organelles do. It just means that there's no membrane inside the cell membrane that houses a region at which that occurs. Okay? They still have DNA, they still have RNA, they still have the capability of making proteins. And in a region which inside the prokaryotic cell, they might have a region called the nucleoid region, but there's no membrane. That's the key component. There's no membrane surrounding that region. Okay, that's what makes them prokaryotic. Eukaryotic, eu meaning true. Karyot still means nut, so true nut or true nucleus. Okay, it has a nucleus, and for the most part, they have internal membrane-bound organelles or compartments in which functions happen. Okay, and so prokaryotics are bacteria and archaea. Eukaryotics are fungi, animals, plants, protista. Okay, so everything else is really eukaryotics. Now, both are really, really important for human health. Obviously, we're a eukaryote, okay, because we're an animal. But bacteria, things that affect us, often make us sick, a lot of them are prokaryotics. So that kind of gives us an idea of sometimes we're dealing with things that make us sick, that have the prokaryotic cell structure. Other times we're dealing with things that can make us sick that have a eukaryotic cell structure. For example, if you have a fungal infection, those are eukaryotes. Okay, and so knowing the difference between the two really gives us a distinct advantage on treating. Okay? If we know that prokaryotes don't have a nucleus and they don't have these organelles, okay, then we can attack certain regions of the cell. Eukaryotes, on the other hand, if we know that they have certain organelles, well, we can attack certain organelles. Okay? And we'll come back and I'll talk about some specific examples of that as we progress. All right, so let's dive a little bit further into prokaryotes. What are we dealing with? So they're the simplest cellular organisms. Okay? They're also the oldest. Okay? So from an evolutionary perspective, they've been around for roughly 3.5 billion years. It's a very, very long time. Okay? They have two main components that we already talked about. Remember we said all cells will have plasma membrane. They have that outside membrane, and all cells will have some kind of internal liquid-based material. Okay, typically it's heavy in water, but also will have sodium, potassium, calcium, proteins, lipids, carbohydrates, all mixed in kind of a solution. All right. Now, outside that plasma membrane, for prokaryotes, they have a cell wall. The cell wall is different than the cell wall that we're going to talk about when we talk about plants, but nonetheless, they have a cell wall. The cell wall is a protective layer, and it protects that plasma membrane. Okay? And then there's quite a few bacteria, okay, mainly in the group bacteria, that have another layer outside the cell wall, which we call a capsule. And this will determine some functionality. These kind of things, like whether or not they have a capsule, what type of cell wall do they have, this will determine our approach when we try to use drugs to maybe get rid of a bacteria infection. If it has a capsule, then we have to get a drug that can get past the capsule. If it has a peptoglycan cell wall, then we have to use a drug that's geared towards disrupting that peptoglycan cell wall. So depending on what the cell wall is made out of or whether they have a capsule will determine our approach to destroying that organism, if that's the purpose. Okay, so the interior of prokaryotic cells, really simple. Again, cytoplasm. Most of the time there's a little bit of protein fibers. Okay, so you got a little bit of framework, but not a lot, because you don't have organelles. You don't have these structures that are membrane bound, so there's nothing really for them to attach to. So there's a little bit in there just to make sure that the cell itself is not collapsing, but again, not nearly as much as you see in eukaryotic cells. Okay? They have ribosomes. Ribosomes are not organelles. Okay? Ribosomes are the site where proteins are made. Okay? And so inside the cytoplasm of the prokaryotic cell, there's going to be lots of ribosomes, lots of places where proteins can be made. Okay? And then they have a nucleoid region, a region where you should be expect to see the DNA or the RNA of that organism or both. Okay? It's not a nucleus because there's no membrane, okay? But nonetheless, it is a region where you would see a chromosome. Okay? So let's look at this, kind of what this would look like. So nucleoid region where you would see a chromosome. Now, the other thing about prokaryotes, they only have one chromosome. Okay, just a single chromosome, and it's circular. It's not linear. Okay, and we'll come back and probably talk about this later. You have the cytoplasm, this jelly-like structure that's in there, kind of liquid jelly-like structure. Lots of ribosomes all over. Okay? And then you have these other things like plasmids, which is another circular chunk of DNA, but much smaller. And plasmids are fairly unique in the fact that plasmids can be determined function of the organism in extreme environments. So a lot of plasmids will have genes that are associated with temperature tolerance or pH tolerance or maybe antibiotic resistance pops up a lot on plasmids. And these are interesting structures because these can be passed to other bacteria. Okay? And part of that passing is the pili or the pilus. These little structures are going from inside the organism to the outside, and it's a connection between the two. These pili can attach to another bacteria's pilus, okay? And they can exchange plasmids. Okay? They can also provide a unit to attach to a wall, okay, or attach to maybe your teeth. Okay, you get lots of bacteria building up in your mouth, and they'll use their pilus to attach to your teeth and adhere to your teeth. So not only is it used to exchange genetic material, it can also be an anchoring point. Okay? And here it's showing you all three options. A capsule, the outermost kind of protective membrane, a cell wall, and a plasma membrane. And then finally, majority of bacteria have some way to move. Okay? And that could be something like this, which is a flagellum. So this whip-like tail, which will allow them to move through liquid. Okay? Some of them use structures that look like piluses or miniature flagellum, and they stick up and they're called cilia, and they can beat those. Some of them can extend their cell membrane, okay, and kind of move throughout the material by doing that. But okay. All right, other structures that can sometimes be found in prokaryotes, and they can relate to all kinds of things. Okay, whether that be locomotion, feeding, genetic exchange, all kinds of things. So again, just recapping on that picture before, the flagellum or the flagella. Okay, most of the time this is used for locomotion, but sometimes it's used for feeding. Okay, and so it can whip material and push material into kind of a feeding groove, and then that material gets stuck kind of in this feeding groove and engulfed and we'll talk about how that works. It's called phagocytosis, when the membrane kind of reaches out around a chunk of material and pushes it in. Okay, the pili or the pilus, again, we talked about that, being able to kind of exchange genetic material. But for the most part, they're pretty primitive. So here's just another example of a prokaryote, and there's not much to them. Okay, a nucleoid region where they're going to have a single chromosome. Okay, a bunch of ribosomes in solution, a cytoplasm, a plasma membrane, a cell wall, and sometimes a capsule, a pilus or pili. Depends also on the organism whether or not they have them. Not all of bacteria have pilus. Okay, a flagellum, ability to move. Again, not all of them have it. Some move in different ways. Okay, but these are just the general structures of prokaryotes. Okay? Next time we come back, talk about eukaryotes.